This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 412, recorded on October 21st, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month, and for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. This episode is sponsored by Drobo, a family of safe, expandable yet simple to use storage arrays. Drobos are designed to protect your important data forever. Visit drobo.com to learn more. Hi everybody, I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi everybody.
Is it raining there?
No, it's not raining. It rained yesterday. And today it's 51 degrees Fahrenheit, feels like 47. I haven't converted that. 11 Celsius.
It was raining this morning here, but now it's clearing up. Mm-hmm. I see some blue skies. The rain has stopped. It's 20 Celsius, 92% humidity. Wow.
Whoa.
It is quite humid here. Also joining us from Western Massachusetts, Alan Dove.
Good to be here.
How have you been?
Doing pretty well. It's cloudy and gray and intermittently raining here. It's 23 C, so pretty warm and muggy feeling. But it had some sprinkles and then it stopped, but it's pretty gray.
It's supposed to rain all weekend here, but we'll see.
Yeah.
Also joining us from North Central Florida, Rich Condit.
Hi, everybody.
Sunny skies, I know. Yeah.
No. We got 80. Well, yeah, pretty much. We got 84 degrees. That's 29 C. Partly cloudy. The dew point is 64. That's a relative humidity of 50%. But I have to tell you that in Austin, Texas, get this. It is 73 degrees Fahrenheit. That's 23 C. 36% humidity. That's a dew point of 44. Sunny. Looking like a perfect day.
Nice.
Well,
there you go.
You'll be there in how many weeks?
Just a week and a half. Actually, we leave a week from Sunday.
Today we have a couple of in-studio guests. We have actually 3. It's filling up here at the Twix studio. We have returning, I think this is his 3rd time sitting in on a TWiV episode, from Drobo, Mark Fuccio. Welcome back.
Hello. And actually, it's the second.
Second only?
Second time online, yes.
The first time you just visited, right?
That's right.
Actually, I think you visited twice before coming online.
Yeah, I think it's become one of the go-to points when I come through the East Coast and I'm in Manhattan.
So you're here for a photography expo, right?
Right. There's an exhibition called Photo Plus Expo in Javits Convention Center. Mm-hmm. It's every October, and I have 2 clients. One is Drobo, and one is another company that hasn't launched yet that is showing its imaging technology through a number of their OEMs and resellers on the floor.
All right.
And Javits, that's going to be pretty convenient to another essential stop if you're into photography, which is B&H's superstore downtown, right?
Absolutely. It's like a 2-block walk, and
the place is like a theme park. It's amazing.
Yeah, it's a cool
It
store.
is. It's designed to be a selling machine.
Yeah.
In the ceilings, they have all the conveyor belts that will move material. So you can go in all the different departments. You can order there. They move it, and you pay and pick it up on the way out. It's truly unique. It's worth a visit.
We have an account here at Columbia with B&H, and you order something, and we get it the next day.
Have you been to the store, Vincent?
I have.
Okay.
It's a little too hectic for me.
Yeah,
it's such a hoot though to go in there and buy any item, and just the whole process of it is just really fun to watch.
The problem is, I don't like being told which doors to go in and which doors to go out.
Okay, yeah.
So nonconformist.
In this store, you have to go in one set of doors and then they say, no, when you want to leave, you have to go out the other. And I just do not like that. I want to go out the same door I come in because they have this flow to prevent theft and all this stuff.
Right.
You know, I don't like it. So I went once with Dixon. He loves it, but I'm not going back. I'm just ordering, and it's going to come. You know, UPS will bring it here, and that's great. So we have two additional guests here in studio. One is, they're both from Ohio, Cleveland, Ohio. And the first is Katreya, who is a fan of the show. Welcome, Katreya.
Oh, thanks for having me.
Am I saying your name correctly?
Yes, it's Katreya.
And you work at Case Western, right?
Yep.
And you listen to our shows?
Yep. I've been listening to TWiV since probably about TWiV 150 or so, somewhere around in there.
Nice. Any of the other shows you listen to?
Oh, yeah. TWiP and TWiM. Started listening to TWiVo, urban farming, so urban agriculture. So
wonderful.
I listen to them all.
What do you do at the Case?
I'm a research assistant in the genetics department in Pete Scacchieri's lab.
Nice. Very good. Well, hope you enjoy listening in. If you want to chime in anytime, you're welcome to. Okay. Your friend here, Molly, unfortunately doesn't have a mic or a headset, but welcome. You're going to have fun with your iPad, right? Yeah, I need to get more mics and headphones. At least one more.
I can see her waving.
Rich says hello. All right. I want to tell you about the graduate program in the Department of Microbiology at the Icahn School of Medicine at Mount Sinai. They want you to know about their graduate and postdoctoral programs. This is a department composed of over 20 virology labs, all centralized in one building in the heart of New York City. This department's a perfect fit for anyone with an interest in pursuing virus research. The department is presently looking to recruit any prospective grad students to apply to their program. The deadline is December 1st. If you want more information about the department, go to bit.ly/micro-MSSM, MICRO-MSSM, all one word. Now, I must say, in full disclosure, this is the department where I obtained my PhD, and you'll find there many, many good virologists, including Peter Palese. Let me scroll down the list of just people that I know. Benjamin Chen, Matthew Evans, Ana Fernandez-Sesma, Adolfo Garcia Sastre, Florian Kramer, Ben-Hur Lee, Jean Lim, who I think Rich Condit also knows, right?
Mm-hmm.
She had Gainesville at one point, is that right?
She interviewed here. Matter of fact, we offered her a job. She went to Sinai instead.
Tom Moran, Beatrice Bogo. Wow, Beatrice goes back to when I was a student. Well, so does Peter, of course. She used to work on pox viruses. You must know that name, right?
Yep. Yeah.
Megan
way
Shaw.
Back.
Megan Shaw. Viviana Simon. Those are people that I know. Ben Tenhoover, of course, who we've had on the show.
Yeah.
And those are the people I know, but there are many more. So if you like virology, you should check it out. And I'll tell you that link again, bit.ly/micro-MSSM. The deadline's December 1st. We'll be telling you about this for a few more weeks. All right, a little follow-up from last week's episode. We have an email from Skip Virgin, also known as Herbert W. Virgin IV. I thought he was the third. Wow. 4 Herberts before him. He writes, thanks.
Well, no, there were only 2 before him. That's why he's called Skip.
Very good. Thanks to you and your team for the kind words about our mouse norovirus receptor paper on TWiV. I remember distinctly seeing a talk from you when I was starting out in which you presented the detailed structure and function of the polio receptor and the interactions between it and the virion surface. I thought at the time that was pretty cool and important. It was really exciting to have an opportunity to do something somewhat similar. The 2 first co-authors are special talents and will go far. Thanks for highlighting them. We have a long way to go with this story. I think that the cofactor story will turn out to be very exciting and revealing. If we can get a grant on it funded, we will see. Be well, Skip. So Skip, of course, was the PI on that paper. Alan, you want to take the next one?
Sure. Anthony writes, the 700 miles covered by a pigeon in one day is for a homer or carrier pigeon returning after remote release. Pigeons don't migrate or take trips on their own. A domestic or feral pigeon will roam about an area of from several blocks to several miles. My guess for the means of the reverse spillover is feral pigeons and wild birds coming into contact with contaminated equipment or litter, and he gives a link to Newcastle disease information. Contaminated equipment or litter near but separate from poultry. That would mean the poultry and feral and wild birds are not interacting directly. If that's so, then the reverse spillover is a curiosity. If that's not so, and poultry and wild birds are mingling, then the discovery of the Newcastle vaccine virus in wild birds is an alarm. Domestic pigeons may be vaccinated for Newcastle. Perhaps they come in contact with ferals, or the ferals are eating discarded contaminated feed. In northern China, the poultry industry mirrors the U.S. One would expect vaccinations and biosecurity. It would be interesting to find out if chickens are vaccinated in the south of China. In that region, poultry producers consist of many family farms raising chickens, ducks, and or hogs. And it looks like, this is a different.
It's also him, but a separate.
Oh, it's also
email,
him. Yeah. Okay, a separate email. Right.
So we speculated about contact with, contaminated stuff. I don't really know. Maybe the authors will hear this and weigh in on what they think is happening.
Well, there are also pigeons raised as food.
Yeah.
And I don't know if there's a lot of overlap between that and chicken farming, probably not in the U.S., but maybe in other places. And I would assume that the ones that are being raised as poultry would probably be getting a Newcastle disease of their own. And I don't know. I know nothing about the food pigeon industry, but that might also be a link.
Mm-hmm.
One of the points of the paper, just as a reminder, was that, if I understood it correctly, is that they don't think that the wild birds are passing the vaccine virus from bird to bird.
Right.
So that means that they're picking it up by some sort of contact with the vaccine contaminated material or birds, direct contact.
So it's ongoing spillovers, not one event. And then Anthony sends subsequent email saying it looks like Bob Dylan predicted climate change back in '64. That should be worth another Nobel Prize. and I won't do Bob Dylan's voice, but this is from the times they are a changin'. Come gather round, people, wherever you roam, and admit that the waters around you have grown, and accept it that soon you'll be drenched to the bone. If your time to you is worth saving, then you better start swimming, or you'll sink like a stone. For the times they are a changin'.
I guess I should have had Kathy read that. She would have sung it, right?
I think we should have made Alan do it in the Dylan voice.
In the Bob Dylan voice. Come gather around, people, wherever you roam. Yeah.
And around you have grown. That's the waters around you have grown.
Admit that the waters around you have grown. Yep.
So did anyone read the Walter Isaacson biography of Steve Jobs?
Mm-hmm.
Yes.
So in there, he tells a story that Jobs was always a Dylan fan, and it took him years to be able to, strike a deal, on iTunes. And one of the things that looped him in is, Dylan wanted the younger generation, to be exposed to his music. So they made a great deal to do it. And then, so Jobs and Dylan got to meet finally for the first time, and they're talking about being older and, health problems and things. And then, Dylan says, well, at least as I grow old, I can still sing like I always have.
I remember the commercial they made with Dylan, right? Of him strumming, and then the iconic people, silhouettes dancing with the white things flipping around. Yeah, that was cool. Neat. Alan, is this you, the next email?
Yes, this is me. So Alan writes, y'all can conclude the you guys arc with this deep dive into the second person plural. And this is something that somebody sent on Twitter, and it's from a site called Atlas Obscura, which is a fun site all around. But they do a very in-depth analysis about a fundamental bug in the English language, which is that we don't have a second person plural.
Yep.
And y'all is a very elegant solution to that. So there you go, y'all.
I don't know if the arc's concluded, though. You'll see.
No, I'm sure it's not, but I was trying. This, though, pretty much covers all the angles, I think.
All right, an update on the contest to win a copy of Viruses by Marilyn Rusink. We still got a few emails of people wanting to be the 50th. That was now 2 weeks ago, 3 weeks ago. I don't know. Steve writes, but send the book to number 25 as they probably need it more than I do. And I can't really sit up to read books these days. Keep up the great podcast. And many thanks for all the help with pace. Steve is from England and writes often. Nikki writes, I would love a copy of the book to read in analog hours. I'm an illustrator myself. The weather here in Oakland, California is, oh, the wind and rain, as the old bluegrass song goes. Grateful for the water, though. Thank you for keeping my brain occupied when I can't have my nose in a book. And Gary writes, hey guys, yes, that is the politically incorrect use of said word. I am hoping to get the book, though I am sure you have already given it away because I had to have surgery and was not able to listen for a while, and I'm still catching up. I'll try to write a longer note later as I have some thoughts and questions I'd like to share and ask. And Cherish writes, hello, TWiV team. I know the contest is over, but I wanted to write you anyway. I don't think I fall in your usual category of listeners, as I am a 4th grade teacher at Riverside Prep Elementary in Oro Grande, California.
Awesome.
I listen to your podcast on my drive to and from work. When you get too technical about the makeup of viruses, it often goes over my head, as my highest level of science education is intro to biology. However, I consider myself a lifelong learner and enjoy learning about things outside my field of study. I must say, I generally do not love viruses, but I do love TWiV. What a great quote. Thanks for creating a wonderful podcast to listen to on my commute. Cherish is a 4th grade team lead at Riverside Prep Elementary. That's wonderful.
Thank you.
Just wonderful.
We will treasure your note, Cherish.
You don't know where Oro Grande is offhand, Mark?
Offhand, no, I don't.
I don't know. I'm sure we could look it up.
But we'll take a look.
All right, we have gotten some haiku limerick entries. They're lovely to the one, and I'll wait a little bit more because it's clear from these last few emails that people have a little bit of lag listening to the show. So we'll give it, a couple more weeks. But I'm telling you, the ones we've gotten are just great. They're just lovely. A very brief Zika sit rep. By the way, I was at a little meeting near the United Nations the other day. It was a Zika meeting. There were 3 speakers. It was sponsored by an NGO. It's involved in health, and I'm not sure what the organization was, but it was in a building that's part of the UN, and I gave a talk, and then there was an obstetrician from Einstein and an entomologist from New York City who does mosquito screens for New York City. Very interesting. And the entomologist said, there is no Aedes aegypti in New York City, and there hasn't been for many, 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 many years. However, there are 51 species of mosquito in New York City, and they collect hundreds of thousands of mosquitoes every season, which goes from April 1st to October 31st. So, April 1st, the mosquitoes all come out. October 1st, they go away.
They go away.
And those species don't include Aedes aegypti, but Aedes albopictus is present in each of the 5 boroughs of New York City. He said there are known colonies of that mosquito in each of the 5 boroughs, and he keeps his eye on them. This year, they've had 3 isolations of West Nile virus, and that's from Culisine mosquitoes, of course. That was really interesting for me to hear.
Yeah.
All right. Our sit rep. What did I do with it? Oh, it's not here. I have to open it up. There are over 150 cases now locally transmitted in the Miami area. 165 non-travel related infections.
Yep.
And they have wonderful graphics, which we've mentioned last year. And you can see...
Well, last episode anyway.
I don't know why I said last year. And there are 745 travel-related infections, 110 involving pregnant women, a little over 1,000 total in Florida.
It looks like that the only recent activity is in the locale in North Miami Beach.
That's right.
According to these little graphics.
Yeah.
The South Miami Beach and the Wynwood things are events there hadn't been any activity in quite a while.
Yep.
And those, the North Miami Beach has also had the least intervention going on. The South Miami Beach, you've got planes and trucks and mosquito pools and all that.
Really amazing, these graphics.
Yes, they're wonderful graphics on this site.
Two other short articles that just came out this week in emerging infectious disease. I just wanted to highlight them briefly. One is called Prolonged Detection of Zika Virus in Vaginal Secretions and Whole Blood. This was a study done at Baylor. It was a non-pregnant 26-year-old female returned from Honduras in mid-May, so this is a while ago. 5 days after her return, which is day 0 in this study, she had signs consistent with Zika, so they began to do a study. By day 17, all her symptoms were gone, but they collected serial specimens for 11 weeks, longer than 11 weeks. Day 0, 2 hours after onset of rash and 2 hours before development of fever, and then 3, 8, 14, 21, 28, 35, 42, 53, 64, and 81 days after onset of illness. They took serum, whole blood, urine, saliva, and vaginal mucosal swabs.
It's sort of peaks in all of the sample areas. There's a bunch in serum at day 0, and that's gone by about day 15. And then in the other sites, urine, saliva, and vaginal swabs, it comes up and peaks around day 10 and is gone by day 20.
Right.
It's gone in the serum by day 15 and persists in whole blood out to 81 days. Importantly, they say that they're detecting Zika virus. They're detecting Zika virus RNA.
Yeah.
Right.
In fact, they looked for virus on day 0, and they got it there by putting supernatants putting specimens into serum, I think, into Vero cells. And they were able to get RNA in the supernatant of those cells. And they also got virus, 8 times 10 to the 5th PFU per mil. But the key here is what was the long-term detection in up to day 14 in vaginal secretions?
14,
right?
Yeah.
And so this is a woman presumably bitten by a mosquito, and the virus is found in many places, including vaginal secretions. Now, this is kind of complementary to a study done very recently in macaques where they intravaginally infected monkeys, and in that case you get also apparent replication in the vaginal tract. And remember, there's been, I think, 1 case of female to male transmission of Zika, so that might explain that.
I wouldn't necessarily call this long term. Her symptoms weren't resolved until day 17. Yeah. Okay, so basically, it's relevant that there's a virus in vaginal swabs, but in semen, as we have seen before and as we'll see coming up,
it's longer.
This can be a very long time. This is
still very
I would call it relatively acute.
Yeah, I mean, I think you need to look in more people to see how long it can go out, but this one isn't very long.
And the presence in whole blood out to 81 days, that's the end of when they were sampling. Yeah. So it could have been longer, as far as we know.
Of course, we don't know if that's virus or bits of RNA, right?
Sure. Mm-hmm.
That's the thing that's always complicated. The other paper is same journal, Emerging Infectious Diseases, Persistent Zika Virus Detection in Semen in a Traveler Returning to the UK from Brazil. So this was a 45-year-old man. He returned to the UK after a 1-week holiday in Rio, got sick a day later with, rash, retroorbital headache, myalgia, high fevers. And they did a number of tests on him. On day 3, they detected viral RNA in urine, but not in serum. 17 days after testing, they found IgM and IgG in his serum. Now, he and his partner were planning to conceive, and they didn't want to wait 6 months, which is recommended by Public Health of England. So, I don't know if this is typical or what, but they did PCR for Zika RNA on serial semen samples to say, okay, if you don't want to wait, let's look for virus, and when it goes away, you could do this. And they found Zika RNA 22, 55, and 92 days after symptom onset. And then no more at day 132 or 174.
if somebody in the U.S. wanted to do that sort of screening, is that commonly available?
Well, you could find a lab that would do it for you. There are commercial laboratories that will do this. It's not going to be paid for by insurance.
Health insurance.
It's not FDA approved, but you could certainly find someone to do it.
But this does underscore that the 6-month time limit is probably a good guideline.
Yeah, for now. I mean,
Conservative.
The obstetrician who spoke at this UN thing said, that's what we have now. As we do more studies, we may change it, may go longer or shorter, but right now it seems pretty reasonable.
Yeah. But,
if this couple said they didn't have sex during this period, but you can imagine some people will, and this fellow, this obstetrician said it's very hard to tell people what to do with their lives. It's a personal decision.
Yeah.
And if they had, they might have gotten infected. He might have transmitted infection.
Yeah, and telling people not to conceive children is a notoriously ineffective public health intervention.
Yeah, indeed.
Once again, this is Zika virus RNA. Not
it
virus.
is RNA.
Yes.
Yep.
Yes, and the number of PCR cycles that they have to do increases over time. So it looks like the amount of that RNA is declining. Mm-hmm. So they have to do more and more cycles, and then finally they can't detect it after 132 days.
I want to tell you about a sponsor of this episode, Curiosity Stream, the world's first ad-free nonfiction streaming service. They have over 1,500 titles, 600 hours of content, founded by John Hendricks of Discovery Channel. And if you know the programming on that channel, you know that you're going to get a lot of real science, not reality science, as you can find elsewhere. It's available. The way you see these programs, you can go on the web, in a web browser. You can look at any of those devices hooked up to the web that interface with your TV, like Apple TV or a device by Roku and so forth. You can see it in 196 countries. They have science, technology, nature, history, interviews, lectures. I just went to the website and searched for science. They have a program called Science Shorts, 23 episodes. Science Breakthroughs. The Science of Walking with Beasts. That was a movie that was pretty cool. The Real Sherlock Holmes. I don't know why that showed up, but I guess there's some science there. Isaac Newton, Exploring Quantum History with Brian Greene. Lots of interesting content. And of course, they have Stephen Hawking's, a number of shows with Stephen Hawking's. And they have a new series called Digits. It's a 3-part series hosted by Derek Muller, who's the creator of the YouTube science channel Veritasium. Explores online safety and security. They also have a very, very large nonfiction 4K library, over 50 hours of 4K content. Monthly and annual plans are available, and they will offer you notable savings. The plans start at $2.99 a month, less than a cup of coffee, for the cost of one title on competing on-demand platforms. Check out curiositystream.com/microbe. Use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 entire months free of one of the largest nonfiction 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign-up. We thank CuriosityStream for their support of TWiV.
And just to clarify something, Hendricks is no longer at Discovery Communications. So people may be listening when you say, this is real nonfiction and not reality TV. And some people do complain about some of the programming on Discovery, but he's no longer responsible for that. He does CuriosityStream now.
He's gone. Huh?
Yeah.
Yeah, but he still was of Discovery.
He was at Discovery, yes, but he's not anymore. So if you have complaints about what's on Discovery Channel and you're wondering if CuriosityStream is the same person, he's not at Discovery anymore.
And you're welcome to complain about Discovery until they start sponsoring us, right? Then you shouldn't complain anymore. All right, I have a snippet which I think is pretty cool. In fact, you'll see later in our listener's pick, it was suggested by Hannah. It is a paper that appeared in Nature Communications. It's called Eukaryotic Association Module in Phage WO Genomes from Wolbachia by Sarah Bordenstein and Seth Bordenstein from Vanderbilt University in Nashville. And I did a TWiV way back, and not so far back, 332, and Seth Bordenstein was one of the guests on that episode. So he talked a lot about Wolbachia, which he works on, and the phage of Wolbachia called WO, or I think we should call Whoa.
Whoa.
Whoa. Whoa phage.
Whoa.
Whoa. I don't know if there are that many O's and H's, but whoa, that would do it. May even turn up in the title of this episode. Who knows, right?
Right.
Wolbachia is an obligate intracellular bacterium. It has to live inside of another cell. It's actually technically an alpha proteobacteria, but you can find Wolbachia in about 40% of arthropods as well as in nematodes of certain types. And we've talked about Wolbachia before because when you put them in certain mosquitoes, it can prevent virus infections. It also causes a distortion in sex. I think you can release females with Wolbachia, like mosquitoes, and then when they mate with males, the offspring die, and so forth. So it's got a number of amazing effects on its hosts. And I read somewhere it could be the world's most prevalent parasite in some ways.
No, I think Dixon has taught us all. It's toxo. Yep. Yeah.
I don't know though. Something that infects insects. It's really hard to beat the insects when it comes to numbers.
A lot of insects.
Okay, I would agree.
A lot of insects, but Dixon's not here, so he can't defend himself. And I know that Katreya, you wanted to meet Dixon. I'm sorry he's not here for you to see him, but you'll just have to come back.
Oh yeah, I'll be back.
You come back in another time. If next time you're back, you can email me and I'll tell you if Dixon's here or not. So these Wolbachias are infected with bacteriophages, as everything on Earth is infected with some kind of a virus. And the particular one that is the subject of this paper is phage WO. And I remember when I talked with Seth on TWiV, he was sequencing the genome, and now he's finished the complete genome of this WO bacteriophage. And he's put it together in this paper.
And this is a little bit tricky because Wolbachia doesn't just grow in regular culture, right?
No, you have to grow it inside of.
You have to grow it inside of cells, so you can't just grow up a vat of Wolbachia and sequence the phage in it. This is a little bit more complex project than that.
So what they did here was to purify the phage particles that are released from cells. But many Wolbachia have in the genome a prophage, an integrated copy of the phage DNA. But in this case, they've sequenced the phages in the genome as part of this study, but they purified phage particles to get the whole genome sequence. The cool thing, what they find, is a huge part of the phage genome. Someone remind me how long this thing is. I can't put my finger on it. How big is the whole phage genome?
The whole phage genome is it depends on which phage you're talking about because they did several.
Yeah.
I think the A1 is, here it is, 65,653 base pairs.
I mean, they find about half of this looks like it's got eukaryotic genes in it. They call it a eukaryotic association module. I don't know why they did that, but they're all in one place in the genome, and there are a bunch of different genes in there, which I'll tell you about in a minute. And they're clearly eukaryotic when you compare them to known sequences. They're clearly eukaryotic genes, which is really the first time this has been seen in a bacteriophage. And when you think about it, the Wolbachia, of course, is living inside of a eukaryote, so this phage has to pass through to get to the cytoplasm of the eukaryote to get to the Wolbachia. So I guess it's likely that it could pick up eukaryotic DNA. So they find,
and it has to invade and escape from two completely different domains of life, passing through into a eukaryotic cell in order to get into the bacterium that's an obligate parasite of that eukaryotic cell to carry out its life cycle. So yeah, you could see some reasons why it might have picked up these genes, but it's still just bizarre.
Bizarre. So they find. Go ahead.
One thing we haven't mentioned, I don't think, is that this is a lysogenic phage, which means that it can exist in the Wolbachia genome as an integrated prophage. Okay, and then occasionally become active and go through a lytic cycle. And if I understand this correctly from the way it's written, the sequence that they had for this phage before came mostly from genomic sequencing of Wolbachia. And they somehow in that missed this eukaryotic association module. And so when they did the sequencing of the phage itself, they picked it up and it increased this 65,000 base pairs, he says, is 23,000 base pairs larger than the previous prophage annotation. So somehow in the prophage annotation, maybe because it was eukaryotic-like, right?
Yeah.
They missed this.
Was it possible that their prophage didn't have this module?
Well, they compare the couple of prophage sequences in Figure 1, and it looks like they've got this module, but those sequences, it's not as big, at least in the scale of this drawing.
Mm-hmm.
I think they went back and sequenced the prophage and found this in it.
They did, yes,
so
they did.
It's certainly, maybe it's not in all of them, or maybe they missed it for computational regions, but it's clearly there.
And in the prophage, isn't it overlapping some other phage genes or phage-like genes, which could have
little
confused
bit,
people?
yeah. Again, depending on the isolate, it does. Yeah,
right.
So they found 10 types of protein domains with functions in 4 different areas. First of all, toxins. This is very cool. I'll talk about that in a minute. Proteins involved in host microbe interactions, proteins involved in cell suicide, and proteins that are involved in secretion of other proteins through the membrane. And one of them that they highlight, and one of the reasons this paper was picked up by the media, is that this phage of an endosymbiotic Wolbachia has a gene from a black widow spider that codes for a toxin. It's called latrotoxin, the C-terminal domain, CTD. And that's the most prevalent eukaryotic-like domain that they find in these prophages. How about that?
Yes.
Black widow spider toxin gene in a bacteriophage.
And this C-terminal domain in particular of the toxin is, what is it? It opens up membranes.
Yeah, it makes pores.
It makes pores in membranes, and so you can kind of come up with a pretty easy idea of what that might be doing for the phage.
Yeah, they point out in this that in order for the phage to get out, it's got to get out not only through the bacterium but through cellular membranes as well. So it's a complicated task.
So the idea in the phylogenetics, when they compare the sequences of this gene, this toxin gene in the phage versus toxin genes that are in spiders, the implication is that it's a horizontal gene transfer between the spider and the phage. And, they say they find, Wolbachia are in black widow spiders. So, and black widows and Wolbachia are in overlapping ecological niches. So apparently the gene has popped over. Now, my understanding is these are open reading frames, right? They're not all mutated and don't function.
Right, I think so.
Right.
So, the assumption is they're doing something for the phage. And what that is, we don't know. But, of course, for this toxin, as we said, it could be poking holes in membranes. That's pretty cool. So they have nice phylogenetic diagrams where, you can see the spider toxin genes along a tree, and then there's the phage toxin gene. You can see they're quite related. Another kind of gene that they find is furin cleavage. It contains furin cleavage sites. So furin is a protease, a ubiquitous eukaryotic protease. And they find lots of cleavage sites for this protease in these eukaryotic genes. And in particular, there's one right before this black widow spider toxin. And they note that this kind of cleavage by a furin protease is needed for maturation and activation of the spider toxin before it kills its victim. So they find a cleavage site for that there. And here's one that I thought Rich Condit would be interested in. They have sequences with homology to POX protein repeats of ankyrin C-terminal domains, and these are called pranks, pulling pranks.
Yes.
And apparently these kinds of domains are also in pox viral genomes, and they're involved in immune antagonism, right?
Right. They mess around with NF-kappa B signaling and the ubiquitin pathway, etc.
question.
Yes.
Do they say anything or discuss how long these genes have been borrowed in Wolbachia? Is it a recent occurrence, or is it something that's been conserved from long ago?
Yeah, so one of them, we'll see in a moment, is thought to be pretty recent. And I don't know really what recent means. You know, when you talk to evolutionary biologists, recent can be thousands of years, tens of thousands, as opposed to millions of years. So I think, I'm not sure what the time scale is, but I think most of them are probably quite a while ago, but they don't really get into that, if I'm not mistaken. So I don't know when this happened. So I would say at least thousands of years ago, but I'm not sure. It's a good question. Some of the other domains they find are ankyrin and tetratricopeptide repeat proteins. That's a TPR. These are repeats that are found in other proteins, and they mediate a broad range of protein-protein interactions, including apoptosis, cell signaling, inflammatory responses, and so on. And here, Mark, is where they say the most recent transfer occurring putatively between the phage and an ant, an ant genome. But they don't say what is most recent. You know, they don't give you a timeline or anything. And
just what, but not when.
Yeah, it is recent, but I don't know what that means. So
right. Well, you'd have to calibrate the evolutionary clock on what exactly. I mean, would it be on the insect time scale, or would it be on the phage time scale?
Yeah, I don't know.
Oh, I was thinking about the transfer of the black spider toxin gene into the phage.
I don't know.
Right.
I mean, you know, on this phylogenetic tree, it's quite distant from the spider venom genes, but there's, as Alan said, there's no calibration of the clock, so I don't know how long it is. We can ask Seth. Yeah, well, that's fine. I'm not sure he's listening, but I can send him an email. Another kind of protein is the NACHT, a programmed cell death domain. NACHT, NACHT. They act as pathogen sensors and signal transduction molecules of the innate immune system and lead to programmed cell death. And these are thought to have come from moths or mosquitoes. And again, a nice phylogenetic tree where you can see these genes in various moths and mosquitoes, and there right in the middle is the phage version of these genes. So there you go. It's a biological computing paper. Rich, what's the right word I'm supposed to use?
Bioinformatics?
Computational biology.
Computational biology. Not bioinformatics, right?
No,
right.
Not bioinformatics.
So the question that's interesting here, many questions, but you know what they do, we don't know for the phage, but how did they get there? So they have three ideas. First, these eukaryotic genes could get to and from the phage genomes during particle propagation in the cytoplasm of animal cells, or during packaging inside of Wolbachia that are lysing and being exposed to the cytoplasm. And so that's possible. Genes may transfer between animal genomes and Wolbachia and then to the phage, and that could happen. Or DNA may transfer first between animals and intermediary entities like eukaryotic viruses or other bacteria and then to the phage. So we don't know. But those are interesting possibilities. I think this is pretty cool.
Yeah, so something just occurred to me now. Yeah. So do I understand it correctly that phylogenetically we have eukaryotic-like genes from the same phage that have lineages that go to apparently different creatures, like spiders or moths or whatever.
Yep.
So what does that say about the history of these things? Does that say that the virus has gotten around, or that they all come from a common ancestor, or?
And maybe the virus has a very broad host range.
Well, it does, right? Because the Wolbachia has a very broad host range, right?
Okay. So the Wolbachia can get around.
Yeah.
So the Wolbachia gets around, it carries this phage with it, and you've got these, these elements that get picked up, and it gets a little of this and a little of that, and eye of mosquito and tongue of spider and what have you.
So we don't have Wolbachia that are species specific for different arthropods. It really has a broad host range. One Wolbachia can get around in a bunch of different species.
I think so.
Not sure.
Maybe
Not certain.
Don't know
maybe
the
we'll
answer
get
to
mail.
that.
Right.
Well, that would make sense. At any rate, that would provide an answer if that's the case.
Yeah.
Yes.
They do. They talk about chlamydia, which are intracellular bacteria, and they're also infected with phages. They haven't found any eukaryotic sequences in those yet, but they're looking. And they end with an interesting question: whether lateral genetic transfers between metazoans and phages are common in the symbiotic virosphere remains to be determined. It's an interesting question, right?
Yes.
So this is cool. This is the first time this has been seen. This a eukaryotic, and a lot of eukaryotic genes within a bacteriophage. So this goes to show you got to be curious, right?
Mm-hmm.
Yeah.
Yep.
Just keep looking.
Now, they make the point in this that this is one of the things they talk about is that this is mixing of genes in one in one virus from a couple of different domains of life, right? Eukaryotic and prokaryotic, and they say that it's not absolutely unique. It's rare because, as we've seen before, some of the giant DNA viruses that grow in amoebae that eat all kinds of different things wind up with genes from different domains of life as well.
Right. Yeah.
This is the first time it's shown up in a phage.
You are what you infect. Yeah.
Yeah.
And this idea of the specificity of the phages in the Wolbachia, that's important. I think we have to get an answer to that.
Yes.
So I'll ask Seth. So was that the only? What was the other question?
And the timeline.
Timeline. Okay, I will ask him.
Yeah. And this, by the way, is an open access article, I think.
It is.
Oh, cool.
In
It
Nature
is. Everyone can
Communications.
see it. You can go and check out the cool pictures of the spiders and
Yes.
phages. The phages are much cooler than the spiders, though.
Yes,
course.
and it's just drawings of spiders in case you're an arachnophobe.
And the model of the different ways that this could have happened is a nice
It's very
final figure of the paper.
Yes.
Lovely. Our second paper is something I've been sitting on for a while. I don't know why. Other things got in the way, but I decided we should talk about it. It's published in Science, and it was published back in October of last year. It's called DNA Tumor Virus Oncogenes Antagonize the cGAS-STING DNA Sensing Pathway by Laura Lau, Elizabeth Gray, Rebecca Brunette, and Daniel Stetson from the Department of Immunology, University of Washington School of Medicine. And this is so cool. It bridges things that we've talked about here on TWiV. And I'm going to just start with this observation, which is if you introduce DNA into the cytoplasm of a eukaryotic cell, if the cell is a normal non-transformed cell, you'll get a innate immune response. The cells will produce interferons because the presence of DNA in the cytoplasm is considered not right. It may be a virus infection. DNA is supposed to be in the nucleus. And the way the DNA is sensed, there are a number of sensors for DNA, but one of the big ones is the cGAS-STING apparatus. And cGAS is an enzyme that makes this very unusual molecule, 2', 3' cyclic GAMP. And the cGAS is the synthase that makes it. cGAS, when it detects foreign DNA or DNA in the cytoplasm, it will synthesize this molecule, which will then interact with STING, and that leads to transcription of interferon genes. And so in this paper, they note that if you do this experiment in transformed cell lines, like HeLa cells, which are transformed by human papillomavirus type 18, they originated from Henrietta Lacks. Remember, she had a cervical tumor, died of it, and the HeLa cells, first immortal cell line, they're immortal because they have oncogene from human papillomavirus type 18. 293 cells, which are human embryonic kidney cells transformed with a bit of the left end of the adenovirus genome, and that contains oncogenes as well. If you put DNA into the cytoplasm of those cells, they don't respond via the STING pathway. They don't make interferon. And so they wanted to know why that is, because if you take, as I said, primary cells, mouse embryonic fibroblasts or macrophages, they will respond to DNA. So what is common among these transformed cell lines?
So you could say that these this is a form of innate immunity. These transformed cell lines have a tolerance.
You could. They don't sense DNA.
To DNA, but what's the mechanism of that?
What is the mechanism is what they want to know.
And a nice control for this is that they show that the RNA sensing pathways, particularly the RIG-I pathways, are intact.
That's right.
So this is specific for sensing DNA, apparently.
Apparently, yeah. Mm-hmm.
I wondered whether if they looked at immortalized cells that didn't have one of these, like mouse MEFs, embryo fibroblasts that were spontaneously immortalized, that didn't have T antigen, what would they see? Would that have been a control?
Yeah, they probably would have seen response to the DNA, I would guess, right? Because they
would predict.
Yeah, and it's a good question. They didn't do that. Yeah,
I guess it would depend on what the defect is in the cell line that allows it to be transformed.
Yeah, I mean the focus here is on viral genes and what they've done, but that's an interesting question.
And they also, I mean, by analogy, the RNA sensors, we know that there are viral proteins that antagonize RIG-I, for example.
Sure.
So what they're angling at here, the hypothesis they're starting with, is that these viral proteins are antagonizing the cGAS-STING pathway.
Right. Now, these cells, the 293s, they're transformed because they contain adenovirus genes, and they produce adenovirus proteins that transform the cells. HeLa cells have the papillomavirus oncogene, and you can immortalize cells by using other viral oncogenes like large T antigen of SV40. And this is why we call these DNA tumor viruses, because these proteins can transform the cells, and eventually they can cause cancer. But their main function, as we've always thought of these proteins in virus-infected cells, is to get cells into mitosis, to get the cells dividing, because DNA viruses, with the exception of those pox viruses, which, as Condit says, don't get involved with the nuclear bureaucracy, they need some part of the DNA synthetic apparatus of the cell. So these proteins are designed to kick the cells into division and allow the viruses to replicate efficiently. And if something happens where the virus doesn't kill the cell and somehow these genes remain, the cells end up dividing forever, and that's why they get transformed and eventually may form a tumor. So they said, does this resistance to responding to cytoplasmic DNA, does it have anything to do with these oncogenes? So they took the first experiment, they took primary mouse embryonic fibroblasts. These are non-transformed cells. If you put DNA into the cytoplasm of those cells, they will respond by making interferon. If you transform them with human papillomavirus E6 or E7, adenovirus E1A, SV40 large T, what happens? So they did this, and it abrogates the response of these cells to cytoplasmic DNA. So it shows you that these oncogenes are specifically blocking the response, and they believe it's the cGAS-STING pathway, which is the main one involved in sensing cytoplasmic DNA. Okay, so that's a cool experiment where they say, all right, it must be the oncogenes that are involved. We'll get back to the original cell lines in a moment. Now,
interestingly, large T knocks out both
yes.
the DNA sensing and the RNA sensing pathways.
Right. Don't
know why, I don't think.
No, we don't know, but presumably they could work on that, right? We'll find out at some point.
Right.
The way these oncoproteins, large T, E6, E7, etc., the way they work is that they interfere with the function of proteins in the cell that regulate cell division, like retinoblastoma RB protein or P53. And it's known from many, many years of work in many laboratories that the oncoproteins bind, for example, E1A and E7 bind RB, and they do so via a very specific 5-amino acid motif, leucine X, cysteine X, glutamic acid. And so you can change this 5-amino acid sequence very subtly, and you can show that that disrupts the interaction of these oncoproteins with RB. And in fact, if you disrupt these oncoproteins in the same way by changing that motif, it blocks the ability of the cell to respond to. It blocks the ability of the oncogenes to prevent cytosolic DNA responses with interferon production. So it's interesting, the same little 5-amino acid sequence that allows these oncoproteins to interact with RB also in some way allows them to probably interact with STING cGAS system.
It makes me wonder whether RB and cGAS ordinarily talk to each other somehow through some other mediator.
Yeah,
right.
It's really interesting that it's the same sequence. I'm really stunned by that. In the end, I want to get back to that and figure out how that happened. Now, if you knock down all the RB family members, there are a couple of them, with short interferon RNA, that doesn't affect the cell's ability to respond to cytosolic DNA. So RB are not involved in that. It's something else. So then they said, what are these oncoproteins interacting with? So they did co-immunoprecipitation experiments to try and figure that out. They didn't find any interaction between the oncoproteins and cGAS, which again is the enzyme that senses DNA and responds by making these cyclic nucleotides. But they did see a good interaction between STING and the oncoproteins. STING is that other part of the DNA sensing molecule that actually senses the cyclic nucleotides and goes on to signal the production of interferon. So the oncoproteins, adeno E1A, and the E6, E7 proteins, I think it's E7 that they looked at, are interacting with STING. Now, back to this 5-amino acid motif that is important for blockade of the STING pathway. Remember, that's a 5-amino acid motif of the oncoproteins. When you change that 5-amino acid motif, not only does it prevent the oncoprotein from abrogating the response to DNA, but it blocks the interaction with STING. They did co-immunoprecipitations to do that. So it's a nice way to tie in this sequence. The interaction of the oncoproteins with STING is important for abrogating the response. So somehow the oncoproteins interacting with STING are blocking its ability to convey the interferon signal in response to DNA. Yeah. Now back to the original transformed cells, 293s, HeLa cells. What if you knock down the oncogene or knock out the oncogenes in those cells? They used CRISPR to disrupt either E1A or E7 in the two cell lines, and in fact, when you do that, they regain the ability to respond to cytoplasmic DNA. So the pathway is intact in these cells. It's just that the oncoprotein is interfering with STING and blocking the production of interferon. So that's basically it. And I think this is so cool because in the end, here's what they say. We have always thought that oncoproteins arose because DNA viruses need to kick cells into division in order to replicate efficiently, right? But they're saying we think these oncogenes evolved due to innate immune pressures on the viruses, and that's why they originally arose. I can't buy that because I can't see having a DNA virus replicating in a quiescent cell unless you kick it into dividing. So maybe they both were selected at the same time because it's the same amino acid sequence on the oncoprotein that's involved. What do you think about that?
Well, this takes me back to TWiV. This takes me back to TWiV 160, where we talked to Pat Moore about tumor viruses. And at that point, and he had published a really nice review that is, I believe, linked in that same episode. Mm-hmm. And he made the point that I really took to heart, that there's actually a significant amount of overlap between the growth regulation and the antiviral pathways.
Mm-hmm. Yes.
And he made the same point that he thought that putting all of the emphasis on these viruses tweaking cellular cell cycle to help them out is probably too narrow a focus, and that a lot of the oncogenes that do that, because of the overlap between the growth control and the antiviral pathways, will have antiviral activity as well. So in this case is apparently different, because it's not obvious how cGAS or STING has anything to do with growth control. Okay, so this is almost like it's very specifically, in this case, focused towards the antiviral pathways. On the other hand, these proteins interact via the same motif with both RB and with STING. So as I said, there may be some cellular, this implies to me that there's some cellular protein that may do the same sort of interaction. Maybe STING, cGAS, it is in the mix having something to do with growth control. Who knows?
Well, I was thinking that this LXCXE motif, it's sticking to some component of the machinery. And if you think about what the DNA antiviral or anti-DNA viral response is doing, it's detecting DNA. And the cellular oncogenes like RB and P53, they are detecting DNA replication and keeping it off. So there's at least theoretically a parallel thing going on here. It's all about detecting and interacting with DNA replication. And it occurs to me that the cell may have hit on some common mechanism that it uses in the oncogenes and in the viral detection system, and the virus is then able to exploit that through a single adaptation.
Yeah, that makes the most sense to me, that a single change took care of both problems, right? DNA replication and innate immunity, not one first and then the other, right? I mean, if they're separate, then I would argue that the DNA replication issue has to be solved first, because I don't see how unless you replicate, but really inefficiently, and you still have to deal with the innate response, maybe that could be it. But I would favor them happening at the same time.
Well, aren't there DNA viruses that preferentially infect dividing cells? I mean, maybe free-living dividing cells?
I think these pox viruses don't need the cells to be dividing, right?
No, they don't care.
They don't care, and probably
they don't care. How about adenovirus, Kathy?
Well, I think it's better if they're replicating. I mean, I think that, that's why there's E1A and E1B to inactivate PRB and P53.
So, Kathy, if you delete E1A and E1B, and you put that DNA into cells, will it go into the replication phase at all, or will it get stuck in early?
Well, it turns out that, now that I think about it, if you have a virus that's deleted for E1A and E1B, if that's DL312, I know it's deleted for E1A. But if you put a high enough multiplicity on the cells, like 300 MOI, you'll get virus replication.
What complicates this, and you know this, Cassie, is that E1A not only inactivates RB, but it releases the E2F transcription proteins that the virus needs for its promoters, right?
Right.
So the idea that a single change in a protein evolved to take care of DNA replication and innate immunity, it's a third thing you have to throw in the mix, releasing the E2F proteins.
Well, the E2F release comes through interaction with PRB.
Right. But that's the same interaction that gets the cells to divide, right?
Right.
So it's three things that are happening. That just makes it even more complicated.
Yeah.
But if we back up before multicellular life,
when would that be? What year, Alan?
Oh, I was awfully young at the time, and I didn't have neurons, so it was hard to.
Okay, go ahead. Go ahead.
But if we go back to that point, I don't know how much we know about viruses of protozoans or whatever was immediately before the first multicellular life, but it would not have had such a problem of encountering non-replicating cells, because if you're single-celled, you don't really need to stop dividing.
That's true. That's true.
So these single-celled organisms would have viruses in them that would still need to deal with some kind of an antiviral response. I don't know what that would look like.
Yeah.
But maybe it looked like cGAS-STING.
Yeah, that's a good point.
And then from there, as we got multicellular life, you might have had a mechanism where the cell needed a mechanism to stop division, and that happened to be the cellular oncogenes that were then simultaneously exploited by the viruses, just coincidentally.
Yeah, that would work. That absolutely works, yeah. If there's a point where you could separate the two requirements, that would be fine.
But we'd have to, somebody would need to look at the viruses of those life forms that are almost multicellular or somewhere in that boundary. Sure. There might be a computational biology project in there if you get enough of those genomes.
There's certainly organisms around that are kind of like that, right?
Yes.
And you could look at that. I'm not sure there's a lot of that going on, but this is the kind of result that would stimulate that kind of investigation. Yeah. Mark, are we making sense?
Yes, you are.
Good. Good to have a little check here to make sure we're not getting too esoteric.
But I mean, this seems very. It seems like very natural that the viruses are evolving to attack different phases of different phases of cells and to take advantage of different mechanisms. There's always some way to get in there and
for sure.
Big or small, and just grab it, reproduce and run.
The cool thing is now, with the tools we have now, we're seeing these issues, right? Which we didn't see years ago. And,
speaking of the tools we have, they talk about this CRISPR knocking out E7, and they say that there are like on the order of 12 copies
That's right.
E7 in the HeLa cells. And I mean, before CRISPR, knocking out even one would have been difficult, but knocking out multiple copies would have been unimaginable.
Yep.
It's incredible.
So when you mentioned, Vincent, the fact that the pathway was intact when they did that manipulation, it reminded me of the work from Edward Goodwin and Dan DeMeo when they put bovine papillomavirus E2 into HeLa cells, and that repressed E6 and E7,
That's
presumably
right. Yes.
all the copies. Then what they found was that the P53 and RB tumor suppressor pathways were completely intact, despite the fact that these cells have, had all kinds of chance to mutate them, and so that just was an interesting parallel to me.
I think they used that in one of the experiments here to suppress E6, E7, and show that you get sensing again of DNA. I think
the bovine papillomavirus E2. I know they acknowledged Dan, but I didn't. They say for the BPV E2 construct.
Yeah.
So was that maybe supplemental data?
Yeah, it's in the main thing. Here we go. We found that HeLa cells transduced with a retrovirus encoding BPV-E2, which silences the promoter encoding E6, E7, also recovered a potent interferon response to DNA transfection.
Okay, there you go.
And it is a supplementary figure.
Yes, figure S2, right. That's right. I think it's a very cool paper. I think it has a lot of cool implications and really neat stuff.
So, question, one thing I was thinking about. So, is there enough body of knowledge? So instead of just doing some sort of computer analysis or reconstructing sequence, do we understand enough about how different nucleotide combinations will work and interact, so that we could actually build a simulator, to simulate how how life or different mutations might evolve, and what behavior that might confer on a cell?
Not yet. I don't think we have enough to predict how proteins or nucleic acids or both would interact in a reliable way. It's one of the in structural biology, people are wanting to get to the point where they look at a protein and can say how it folds, right?
Yes.
And so we're not close to that either. But at some point, we may have enough data that we could do it.
So is that Vincent? Is that a problem of not having enough computational cycles, or do we just don't have a well enough developed theory?
No.
Yes.
Which one is it?
Yes.
Yes.
Both.
You need at least one. If you don't have a really comprehensive understanding of how proteins fold, which we don't, then you would need an immense amount of computing power to kind of semi-randomly figure it out. Well, the main approach that's being used now is training machine learning algorithms on here's a sequence, here's what the structure looks like, here's another 300,000 sequences, here are what the structures look like. To do this, you need an enormous amount of structural information about a family of proteins. And it turns out that for families where you have lots and lots of structures, especially soluble proteins, because they're relatively easy to crystallize, you can teach algorithms to make pretty good predictions of at least things at the domain level and working up toward the protein level. In areas like multi-transmembrane spanning proteins, where we don't have a lot of structures because they're really hard to work with, there is not such good success. So it depends a lot on what kind of protein you're looking at. We don't fully understand from first principles what would be causing this, and that is the other part of the problem. But it does take an immense amount of computing power to do these kinds of calculations too.
Right. That's why there's the folding at home.
Yep.
Folding at home is
Exactly.
one way of, yes.
Exactly. Well, I'd like to tell you about a sponsor of this episode, which is Drobo, and I feel funny doing a Drobo spot with you sitting here. Do you want to do the spot yourself, Mark? Or should I go ahead and you can interject?
I'll just make a comment at the end.
Okay. Our sponsor is Drobo, the makers of safe, expandable, simple-to-use storage arrays. These are arrays where you connect them to your Mac or your PC using Thunderbolt or USB 3. And they come in different sizes from 5 drive bays to 8 to 12. And you can use them to store your files and do a variety of things. And what's cool about them and what's unique is that they're designed to protect your data, your important data, forever. No other storage array has such a high goal. And I love the story, Mark, that the founder of Drobo originally lost pictures of his honeymoon, and that spurred him to find something better than RAID, and he came up with Drobo. Beyond RAID, which has a lot of unique aspects that you don't find in regular garden variety RAID. So what's unique about Drobo? So first of all, they protect your data. And in the very beginning, the idea was to protect against a single drive failure. So if you have 3 or 4 or 5 drives in your Drobo, if one of them fails, all your data are protected, because the way the array is designed is that if you fail a single drive, you can simply replace it and the data will be reproduced. From the very beginning, there's been a battery-backed cache in the Drobos to store any data from the computer in case of a power failure. That there's no time to write it to disk, it gets stored in this cache. We now have dual drive redundancies. You're protected against 2 drive failures, and you're also protected against the random bit flips and corruptions that occur with data on a daily basis. And your data are protected forever, essentially. The Drobo will go on beyond your lifetime and protect your data beyond the time that you're around to even look at it anymore. Increasing capacity. You can add more and more drives to your Drobo. If you have a 5 drive bay Drobo and you're filled up, you can increase the size of the drives up to, what's the biggest drive you can buy now, Mark? What size is it, 4 terabytes?
Oh, no, it's gone way beyond that. It's sort of the largest in commercial mainstream is 8 terabytes,
Wow.
and Seagate is starting to sample 10 terabyte hard drives.
Wow.
So
That's amazing.
It's amazing, yeah.
10 terabytes. And so, I have 2, and I think that's really great, 2 or 3 terabytes. But you can just increase your capacity, and it's as simple as taking one drive out and putting a bigger one in. You can have mixed sizes in the array, and you'll instantly have different space. And, of course, this is all simple to set up and use. It's the use of a Drobo on a daily basis is based on feedback from lights that are on the front. Next to each drive bay, there's a colored light. If it's green, it means you have plenty of room. If it's yellow, you're starting to fill up the capacity. You should think about buying a bigger hard drive or adding a new one. And if it's red, it's full. You simply take out the drive and put in a bigger one. And, of course, if the drive is flashing red, your drive has failed. But even then, just pull it out, recycle it, of course, and put in a new one, and you're ready to go. Designed to be simple to use. If you can read a stop light, you can use a Drobo, and of course Dixon has one from years ago, and he's still using it. And I know Dixon is a little bit technologically challenged, and he never has problems with Skype. But he never has problems with his Drobo because it's a great system.
So I've had a question for a long time. I consider myself a fairly small user. For instance, maybe on the order of a terabyte is enough to store everything I have. And so something that Mark just said maybe answered part of my question, that this works with whatever hard drives I have, whoever I buy them from. I don't have to buy Drobo brand hard drives.
Drobo
right?
doesn't make drives. No,
That's what I thought.
It works with the leaders in the market now are Western Digital, Hitachi, and Seagate.
Mm-hmm. And I have some of those. Yeah.
Yes, and they all work. So I think Kathy, I detected maybe an implicit question of, is Drobo for me? And and I'd say certainly Drobo's for everybody, but I think really the touch point is a simple test: does your data fit on a single hard drive? If it does, then Drobo's probably not right for you. It would be a more expensive solution and hence overkill. But if your data won't fit on a single hard drive, then I think that's the sort of person who should start to think about having a Drobo.
Well, or if you're just the belt and suspenders type who thinks you can't have too robust a backup, that might be a good market.
That's right. Yeah, that's another one. Because, one of the things that, again, was a problem of a traditional RAID is when it does when a drive fails, you have to rebuild. And that will put all the other drives under additional stress. And a problem that's very common is if one drive fails, if the other drives came out of the manufacturing lot, studies have shown they're statistically likely to have another failure. So if another drive fails while it's doing a rebuild, boom, kiss your data goodbye. So Drobo accounts for that in two ways. One, it can protect if you have three or more drives. You can protect against two drive failures, and it's not that you're worried about two drives going bad at the same exact instant, but that you don't want to have that second drive fail while you're doing a rebuild. And the other benefit about sort of the rethinking of RAID, and that's why it's ultimately called Beyond RAID technology, is RAID originally was developed for the enterprise, and there they have capital budget cycle, so they'll buy it, they'll put the drives in, they'll start, it will be mostly empty, and it will get full over 3 to 5 year, whatever their capital budgeting cycle is. Normal people don't do that, and they have 3 terabytes of data, so you get a Drobo, get 2 4 terabytes hard drive, put it in, and then as you collect more movies or take more photos, you need more space, and then get another hard drive and put it in. So that's why it's important to be able to have number one, the capability just they had a hard drive at any time, because that's how most of us work, right? And then number two is support mixed capacity hard drive. And traditional RAID, the available space is a function of the smallest drive. So if you had a 1, 2, 3, 4, 5 terabyte hard drives, all the space, you would be having 3 terabytes of protected data. With Drobo, you'd have 1 plus 2 plus 3 plus 4. So what's that? 10, if I've done the math correctly, of available space. And very, very, very simple idea. And I think maybe for the interest, some listeners of the show, it really shows sort of what your starting goals and objectives are, leads to what the system is. Mm-hmm. And there is no sort of teleological sense of how life emerges. There's no I'm getting myself in trouble, but maybe there's no evolution is dumb and harsh instead of having intelligent design. But with Drobo, there was a ability to look at 20 years of what are the limitations of traditional RAID, and that's what led Jeff Barrell to when he was looking for a solution. He basically said, "Yuck! I don't want to manage this." He developed high end network attached storage systems that sold for over $100,000, and last thing he wanted to do is have to manage that complexity at home. So,
right.
That's long story short of how that new technology came to be.
Okay.
Well, if your interest has been whetted by Mark, you can save $100 off your purchase of a Drobo 5D, 5DT, 5N, or any 8 or 12 drive system. Just go to drobostore.com and use the discount code microbe100. We thank Drobo for their support of TWiV, and thank you, Mark, for helping bring us together.
Thanks for allowing me to visit.
Oh, anytime. In fact, if CuriosityStream wants to visit, they're welcome too.
Yes.
Tell us all about it, but I have a sense that they won't. All right, we can do a few email here. Rich, can you take that first one since you seem to have done a bit of work on it?
Yeah, I got into this. Steve writes, "Hi, Vincent et al. I was rather surprised to see this most unwelcome report in the retraction watch lists yesterday. I've been quite diplomatically trying to reassure friends and contacts that media fears about the various HPV vaccines were unfounded. And I don't know if he enclosed a Retraction Watch link. I looked at Retraction Watch and couldn't find anything relevant to this, but apparently it's something that alerts to some fears about side effects of HPV vaccines, which we'll get into. It now appears that there were conflicts of interest amongst the EMA review team, that's the European Medicines Agency, and that the manufacturer's assurances were given precedence over those of experts in the field in Denmark, and also, among other things, placebo vaccinations with aluminum adjuvant were compared with complete vaccines, thereby hiding any effect of the adjuvant itself. Concerns were raised about the unreliability of the reports provided by the drug company, were raised by two co-rapporteurs, but overruled in the final report by the rapporteur who Cochrane shows has in fact had links with some of the drug companies. Public and secret versions of the final report were made and all participants were sworn to secrecy. I don't pretend to follow all the intricacies, but it rather looks as if the anti-vaccine lobby has just been handed a mighty bit of propaganda by the secrecy and duplicity of the European Medicines Agency. Regards, Steve, who is in Luton, Bedfordshire, in England. So, as I understand it, this originates from a Danish study where some neurologists had a bunch of patients exhibiting one of two different syndromes, one called POTS, postural orthostatic tachycardia syndrome, which means your heart starts beating really fast when you sit up after lying down. Another is chronic regional pain system, or CRPS, and that's, as it states, chronic pain in your limbs that apparently can be pretty debilitating and go on for a long time and become worse and spread. So these are both neurologic syndromes. They could theoretically have an autoimmune component to them. So one starts to think about a vaccine inducing an autoimmune response, as we have discussed before, but that's just an idea. At any rate, in this Danish study, there were something like 53 patients with these symptoms that the study, the people who ran the study noted had all had an HPV vaccination in the same sort of time period as they first came up with these symptoms, and so made that association. In those publications, the Danish authors say that they understand that this doesn't mean that there's a real association between these syndromes and the vaccine, but they're just pointing out that there is apparently a correlation in time, and could we look into it? And apparently they appealed to the European Medicines Agency, which looks into this kind of thing, and the European Medicines Agency launched an investigation and came up with a report, and the Danish group, now backed up with a larger organization. It's the Nordic or Scandinavian arm of the Cochrane Group, which does meta-analysis of various medical studies, came back and very sharply criticized the EMA report. They felt like they just kind of blew them off and didn't give the observations a really fair review, and found, as is listed in this letter, a number of different problems. So this is an ongoing objection. I note at the same time that this association has been made elsewhere. I wrote Paul Lambert with this, asking if he knew anything. He said he was not very familiar with the story, but he was familiar with something that sounded similar in Japan. And indeed there has been. And in Japan, they even decided, because there was a media storm over this, decided to no longer recommend the HPV vaccine in the normal childhood vaccines. And so
that's bad.
It's now a more, that's not good. So the way I see it, this is an ongoing thing. The Danish study and a couple of other observations have made this temporal association between these syndromes and the HPV vaccine. If it does occur, it's rare, like 1 in 100,000 or something like that. And their EMA looked into it, but there were flaws apparently according to these individuals and Nordic Cochrane in their approach to it. So it would seem that this is something that needs further, that could benefit from further investigation. It's, in my opinion, it's easy to blow this kind of stuff off, but I think you really have to take this kind of stuff seriously and track it down, and hopefully that will be done. If there's a problem, it's rare. Nobody argues with the notion that the benefits of the HPV vaccine still vastly outweigh any risks, even if this association is real. But it's something that probably could benefit from some more investigation.
Yeah, the Danish work seems to be exclusively in patients who received Gardasil. I don't know if that's because of the way that country has its vaccine program structured or what. And so there are 2 different HPV vaccines on the market, and it may be that they differ.
They differ in their adjuvant.
Right. It may be. Okay. It may be that the different adjuvant is having an effect. There's a lot that could be going on here, and it's very spotty evidence. But I agree with you, Rich. This is certainly something that needs to be investigated and taken seriously, if only because you want to have full confidence in a vaccine like this.
Absolutely.
Rich?
Yep.
Did you say what the incidence rate was of these complications?
What I get is about 100. The numbers that I've seen is about 1 in 100,000.
Which is
so it's 0.1%, if I've done the math right.
Right.
It's low.
Yeah. But bear in mind, this is based on a few dozen case reports from possibly 2 groups, and I don't have references for the Japan research. The 2 studies from Denmark are both from the same group. Right,
and we're talking about 53 patients.
Right,
so
this needs to be reproduced by somebody outside the groups that. I'm not saying that they did anything wrong, but it needs to be shown that this is an independently reproducible phenomenon, and then proceed from there to figure out what's going on, if indeed there is anything going on.
Yeah, the big flap here is that the Danish group objected to the way the EMA handled their request to follow this up.
But I think their complaint is a little over the top, because this is essentially one report. I mean, yes, they have 2 papers, but they are one research group that is spearheading this claim, and they must understand that this is something that needs to be validated before you go telling millions of people not to get a vaccine against something that could kill them.
Yeah.
Right.
You remember the CDC has a adverse effects reporting system in place for this vaccine. When they've collected a lot of information, and you can find it on the CDC site, and they've said they had 80 million doses of HPV vaccine distributed in the U.S. since 2006. The most common side effects include pain, redness, and swelling in the arm.
Now, VAERS, of course, it's a problematic system because you can do all kinds of abuse with that, and the anti-vaccine folks do. But that would be the starting point for a study to try and follow up and see if you see. I'm presuming the CDC has already gone into VAERS and searched the database for this cluster of symptoms and would then try to follow up and conduct some kind of a study there.
Yeah.
I put one link in here to a Medscape article that's it's over a year old. It's July 2015, but it summarizes this pretty well.
All right.
And it's behind a paywall.
Yeah, Medscape is a problem, yeah.
Okay, all right.
For Medscape, you have to register, though, right? That's all you need for that?
That's what I did, yeah.
Okay.
Although I was annoyed, so I didn't.
Yeah.
But that's just me. That's just me.
Or you should do as your son says and use some fake email address.
Throwaway account, yeah.
My son has many, fake emails that he uses. Yeah, smart guy. The next one is from Suzanne, who sends a link to a... it's actually something we've probably had before. It's an interview with Brian Deere by Russ Roberts about his investigation of Wakefield, which Suzanne thought of when we talked about it with Nina Martin a couple of months ago. Yeah. The next one is from Jean-Michel Claverie, who one of the discoverers of giant DNA viruses. And so we covered this new thing called Mimivir on TWiV 380.
A great episode.
Remember that, Mark?
Yes.
And the name is actually quite...
Well, I don't know if I remember the episode, but I remember all the large viruses. Yeah.
TWiV 380 was called Viruses Visibles in le Microscope Photonique. Even gave it a French name. So it's about this thing called Mimivir. And Jean-Michel saw it and he wrote, Mimivir is one more nonsense published by Nature. See enclosure, published there because of the censorship of Nature. The authors have basically to agree with a rebuttal for it to be published. So he sent me a critique of the original Nature paper on Mimivir, and he had to publish it in a weird place. I don't recall where it was right at the moment, but he said, if you want to criticize something in Nature, the authors have to agree to your criticism before it can be published, which seems weird. Right?
Yeah.
But anyway, here's the upshot. Jean-Michel and Chantal Abergel are right. The original observation is that Didier Raoul felt they discovered a CRISPR-like DNA-based defense system in mimiviruses, and that this confers resistance to virophages. And we talked about that on TWiV. I was frankly very excited. They examined the sequences of lots of mimiviruses. They found a 28 nucleotide sequence identical to a virophage, and these lineages are resistant to the virophage, so they said it's a CRISPR system. And Claverie and Abergel think they're wrong, and I'll put a link to the paper. Let's see if I can tell you right now where it was published. It was published in Virologia Sinica. Okay, so he's a little upset of having to publish there, but as he said, he couldn't publish it in Nature. They wouldn't let him. They think that they're dealing with some kind of defense, but it's a protein-based defense system. And they have three reasons why this is true. The first problem is that mimivirus and Zamiome replicate in the same location, making a CRISPR-like defense system difficult to conceptualize. The second problem is that Zamiome sequences in the mimivirus genome are not regularly spaced or flanked by recognizable repeats. And finally, they think there's no way for the nucleic acid to distinguish between the virophage and the sequences in the mimivirus genome. Right? It's going to degrade both of them. So they think there's a cellular... In fact, they think that this is part of a protein-based system, which, of course, would have to be proven experimentally. And I think somewhere the original authors of this Mimivir paper responded and said, "Yeah, you're not entirely wrong. We'll work on it." So it's an interesting story, and we'll see what happens there. Where are we now? We finished with Claverie.
Now we're down to
Alan.
PB supernova.
Okay. Alan, well, let's see. Have Kathy take that next one, please. PB supernova, yeah.
Hi, TWiV peeps. I'm a postdoctoral fellow studying reproductive biology and endogenous retroviruses. I'm also interested in interactions of exogenous viruses with cells in the reproductive system, but I'm not a virologist. I'm in a physiology department, and at my current institute, there is limited virological collaboration potential. My question is this: what is the utility of an organ-specialized or even cell-specialized virologist? For example, I'm interested in viral infection in testes, spermatozoa, Leydig cells, etc. What are the job prospects for someone like this? What department should I consider when applying for jobs, the reproductive side or the virology immune side? My second question, after listening through your archives, I was fascinated by the Rocky Mountain Laboratories, where it is currently 66 Fahrenheit, 18.9 Celsius, and sunny. How does one get a PI position there? Sincerely, PB Supernova. And this was probably written a while ago. It's probably not that in Hamilton today.
Nope.
No.
So what is the utility of an organ-specialized virologist or cell-specialized virologist?
I think you just need a question, right? You need to have something you want to answer. It doesn't matter where the infection occurs. I mean, if you want to say, how does Zika replicate in the testis, that's fine. But, I think more generally it's not great because you need to have some kind of problem that you're trying to solve, right?
Right. I agree.
But it could help to have a specialization in one tissue or organ.
Yeah, for sure. Yeah, but so this person is interested in these reproductive cells, and there's certainly a number of viruses which can go there. But just being there is not enough. You have to have a problem. And, if you want to look at transmission, sexual transmission of Zika, sure, you could do that. But it's got to be based in something. I would say how to get a job at Rocky Mountain, a PI job. Well, that's tough. For first, you have to...
It's like, how do you get to Carnegie Hall?
practice. You have
publish.
I mean, you have to get a PhD, or you're a postdoc. Yep. Okay, so you want to get a PI job. Well, you could write, and, I know I did a podcast there a couple of years ago, and what's the name of my contact there?
Bloom.
Marshall Bloom. And you could just write him and say, I love you, and can you give me a job or something? Or write and see if they have any positions.
Marshall's a pretty easygoing guy. That'd work.
But
well, I think that, too, should start with a slightly different premise, which is not, gee, it looks nice there. I'd like to work there. But
yes.
What is it you're going to bring to them?
Yeah, they need certain things.
Why do they need you? And then take it from that angle.
I mean, they tend to have people working on relatively dangerous things like Ebola and MERS and tularemia, because it's a BSL-4 laboratory, so its specialty is that sort of dangerous stuff.
Right. So start a project on Ebola virus and semen, and then try to leverage your way into RML that way.
They also are very interested in ticks. They have a big tick collection program there, and and it's beautiful there. You can go out and collect ticks and look and see what's in them.
It's beautiful. Forth. You can go out and collect ticks. Yes.
Yeah. I mean, it's a beautiful. It's the Rocky Mountains. You can not just collect your ticks, because people do that here in Connecticut and New York State. It's not as pretty. And obviously, that doesn't matter. But I was impressed by the surroundings, and I talked to people, and they say, "Yeah, we love going outside and doing tick collecting because." It's really nice out here. But you'd have to have something that they're interested in. They have a mandate. It's a government laboratory, and they have to fulfill their mandate. So you should check that out.
Where was this? Hamilton?
Hamilton, Montana. I know that Dave Quammen said it was starting to snow in the mountains, right, Aiden? And Alan, a couple of weeks ago, right?
That's right.
So it's getting chilly.
It is currently
your altitude.
Yes.
It's currently 61 and partly cloudy.
I'm going there on November. When am I going? November 17th, 18th.
My guess is it won't be 61 and partly cloudy.
Right.
Yeah, but it's not going to have a lot of snow in the valley there. Where
in the valley, exactly. If you go up the mountain 10,000 feet higher or whatever, then it certainly should be snowy.
So I'm going to do a TWiV there at a local high school, because there are a number of technicians at the lab who graduated from the high school. So we're going to get them and some of their science teachers from the high school and have a little conversation.
Fun.
And that could be fun.
That's great.
So that should be fun. We have a question from Don, who wants to know if we've ever examined the English sweating system. Sickness. Sorry. And yes, it was so long ago we talked about this on TWiV, and I don't remember the episode, but is a kind of a long involved thing of a weird illness that occurred first in England in the 1500s and then something similar in France. And I found a wonderful open access research review where the English sweating sickness and the Picardy sweat caused by hantaviruses. So people have thought of a lot of different agents that might be involved in this. And of course, it's so long ago that who knows? Yeah. The best part of this article, the conclusion, it's evident that all arguments without evidence for the causal agent of English sweating sickness or the Picardy sweat is mere speculation and circumstantial evidence. And he quotes Sherlock Holmes, "Once you eliminate the impossible, whatever remains, no matter how improbable, must be the truth." So I don't know. Anybody want to chime in on English sweating sickness?
Looks like you discussed it on TWiV 167 in the letters.
How did you find that? You just searched TWiV sickness sweating?
I Googled, yeah, exactly.
Oh, okay. Alan, can you take Islam's, please?
Yes. Islam writes, "Dear Professor Racaniello and Alan, thank you very much for the quick chat we had right after TWiV at ASM yesterday." This is from, that was back in Boston, I guess.
That was in June, right?
In June, yeah. This is, that was a while ago. Briefly, I'm a virologist currently working at MIT. I have a DVM and MS degrees from Egypt, then moved to Cambridge University for my PhD. I was a postdoc at the NIH and KU before moving to Boston. More info about my background on my personal website islamhussein.com. I was just focusing on my research in the lab until about 2 years ago when the Egyptian armed forces came up with a huge fiasco about 2 devices for treating and diagnosing pretty much all viral infections. I was so frustrated by that and wanted to explain to people why this is wrong. I couldn't compile my thoughts into an article or a Facebook post, so I decided to record a video and publish it on YouTube. The video went on for about 80 minutes, and unexpectedly it went viral. My story was covered by Science magazine, and he gives a link to the article. This is what got me into science communication. Since then, I've written several blogs and was also hosted by some Arabic TV channels to comment on a variety of viral diseases. I decided to start Viral Vlog, VIROLVLOG, which was inspired by TWiV but geared towards an Arabic audience. I recorded a few videos as screencasts and published them, but it turned out that Egyptians were not happy with voice-narrated PowerPoint slides only. They wanted to see my face. It seems these humble efforts made a difference with a large number of people who were hungry for knowledge about viruses but were not comfortable enough to digest the widely available English content. Nature Middle East has recognized my work, listed me as one of top 6 Arab science communicators, gives a link to that. With help from my 12-year-old son, Adam, I had the experience of recording videos for Tahrir Academy, an equivalent of the Khan Academy in Egypt, in my home office. I used to record raw videos and send them via Dropbox to their professional team for editing. With Tahrir Academy, I produced a 4 video series on influenza, Ebola, critical thinking, and MOOCs,
Massive online open
courseware. Last year, I gave a talk entitled Alice in Viral Land at TEDx Cairo, and the response from a young audience was overwhelmingly good. And Tahrir Academy, as an NGO, non-governmental organization, was discontinued, so I decided to move down to the basement and establish my own self-funded semi-permanent home studio. Adam and I have also learned the basics of video editing, started producing our own vlogs, made at home all the way. We discuss several topics, including discovery of viruses, influenza shifts and drifts, how this affects vaccine production, viruses, are they alive or dead, Zika virus, CRISPR. I also started recording interviews with Egyptian scientists in all sorts of fields and tried to connect their work to viruses. So far, I've had an episode with an Egyptian geologist who was a member of the NASA Apollo team. We talked about viruses in space. I also discussed the history of quarantine with an Egyptian historian at Harvard. Pending an approval from Google, my next video is going to be recorded inside their office here in Cambridge, and we'll discuss how computer scientists picked up the words viruses and viral. We will also cover digital epidemiology and touch on Google flu trends. Collectively, my videos have reached more than 600,000 views so far. They're penetrating way beyond Egypt to all Arabic-speaking countries. We're learning a lot from the feedback we're getting from our viewers. I still have many other topics to cover and will continue this effort as a hobby side by side with my job as a full-time researcher. And then he provides links to the Viral Vlog YouTube and Facebook pages and requests our help in in promoting this. And I will say, Islam, I have been meaning to make this a pick of the week for quite a while.
Yeah, we had told him we would do that.
Yes, we said, yeah, we'll pick that on the show. So, we do eventually get around to these things. Now, of course, if you go to this Facebook page and the YouTube channel, it's all in Arabic, which
that's the point, right?
Is really kind of the point. And when I was talking to him at ASM, I said, this is awesome. We really need more people doing this kind of content in other languages because, I can kind of barely get by in Spanish, and that's about the only other language I can do.
I barely get by in English.
And so it's really, really wonderful to see a project like this and to see that it's taking off. And if you speak Arabic or read Arabic or are interested in brushing up on your Arabic, point yourself or your friends to this YouTube channel and Facebook.
And Mark, if you want to sell Drobos there you go.
Yeah.
Get on this guy's show.
Oh, that's interesting.
I don't know. Do you sell overseas at all?
Yeah, we sell through Europe. There's an online store. It covers a lot of Europe and the Middle East and presence in Japan, China,
right,
Korea.
Yeah.
I want to tell you about the 2017 ASM Scientific Writing and Publishing Online course. This is an interactive webinar that explores the scientific and writing and publishing process with ASM Journal's editors. It'll cover titles, abstracts, figures, legends, the manuscript, and the whole review process. It'll take place from January through April of 2017. You have to register, and there's a little charge for this. And I think, if you're a student or postdoc, you should ask your PI to pay for it. The deadline is December 1st. You can learn more at bit.ly/SWPOC17. I think it's $150 or something like that. People should be willing to pay for you to take that. All right, let's do some science picks of the week. Mark, do you have something for us?
I do. I have two. One I just jumped to here, just listening to some of the question about careers. And, one thing that maybe for some of your listeners, if they're still in graduate or undergraduate school, if they're interested in maybe a career in industry instead of academia, is if they're interested in startups and maybe that way of going. There's an organization nationwide called EFN, Entrepreneur Futures Network, and they hold contests to help students, and they provide mentoring sessions to help students understand what it's like to be an entrepreneur and target a market, target a problem, and so on and so forth. And I've helped mentor a variety of things out in the Bay Area for it. And their website is enterprisefutures.org. So that may be another resource just for people to think about how to use their knowledge and how to apply it in different fields. And the second was more of a sort of an interesting thing, and I recommend a podcast that's called Mission Log, and it's a Roddenberry Star Trek production, and it's two co-hosts, John Champion and Ken Ray. What they do each week is they analyze an episode of Star Trek, and they take one episode per week, as well as all the series and movies, and I think they have job security until probably the year 2024 or something. But
until the date of the actual start of Star Trek, probably still Stardate, whatever. That's
right, and it's interesting because they take each episode apart, and they try to look at messages, morals, and meanings, and many of the Star Trek episodes deal with things, combinations of virus, parasitism, and
right.
There's other interesting subjects on it. So it may be time consuming because you may want to watch a show on Netflix, and that's an hour, and then listen to the podcast. On lunch is on Thursday, so that's another hour. But
it's a weekly podcast.
It's a weekly podcast, yes.
Cool. Yeah.
And it's Mission Log Pod, and I've gotten to know them and Rod Roddenberry a little bit, who's a son of Gene, the creator of the series, and it's a really great show. They show a lot of behind the scenes scripts and documents and photos on the website, so it's an interesting way to get behind some of the backstory of the series and episodes.
And that's because the sun is got access to all of that, right?
Oh, indeed, yes.
Yeah, that really helps. Of course, we have access to viruses. That's why we do TWiV, right? That's
I
neat.
get lost in this.
Yeah, I can see you doing that, Rich. Are you a Star Trek fan?
You bet.
Yeah.
Which series do you like most?
Well, I think Next Generation. I really like
TNG.
that.
Okay.
So I must say that I haven't I haven't paid as much attention to many of the other series. How about you?
Oh, it's probably either the original series or the Next Generation.
Yeah, Next Generation is just great.
Well, since we have you talking, Rich, what do you have for us? Thank you, Mark.
Thanks.
I just stumbled across this thing. Oops, I better silence that. All right, called the Zuta, Z-U-T-A. It's a portable robotic printer. So this is actually still in development. You can pre-order one. The link I put is not to there is a website for this thing, but the link I put was to a video that describes it that was produced by somebody else, but it's a better description of what it is. This thing is small enough so that it'll fit in your pocket. It looks like, maybe the size of a couple of computer mice. And you just set it down on a piece of paper and send it a wireless print command from your phone, and it crawls around the paper and prints.
Oh, this is adorable.
Yeah, my brother showed me this. But you're right, Rich. This is a cool video at the Henry Ford in Dearborn. Yes. Very nice.
Yeah. And the nice thing about that is it describes more how it looks. It also has a picture of it with its clothes off so that you can see what the inside looks like. And so I thought it was pretty cool. It looks annoyingly slow relative to what we're used to now, and it's only black and white, but my guess is this is only the beginning. Okay.
I thought we're supposed to get away from printing, from killing trees.
Well, yeah, I suppose, but every now and then you have a need.
It's pretty interesting. Huh. Well,
and Rich, when you hesitated on the plural of mice or mouse for computers, I was just listening to an older episode of Away with Words, and someone asked that very question. And evidently, mouses is used in the tech community, but mice is still more prevalent.
Okay.
But it was kind of interesting because then they made the point that the computer tower or box that the big old computers used to be, the plural of that is boxen instead of boxes.
Yes.
So that the tech community has this interesting way of making plurals, so mouses is okay.
Hey, if a trilogy is 3, what is 2?
Biology.
Biology. I've been listening to this on a podcast recently, and they can't decide what 2 is.
Y'all.
Y'all. That's pretty funny. Kathy, what do you have?
I picked how Lego helps blind people see. It's a Braincraft video that I got from a completely different route rather than just Alan's Braincraft pick from last week. But she goes to Boston, and you see her going on the train. And anyway, she interviews a blind person, and he and another person together have worked out verbal instructions for how you put Legos together so that blind people can do this. And as she said, reading those instructions would be, if she couldn't figure out how to put a Lego together from reading those instructions, but for somebody blind and listening to them, that's the way they can do it. And then they make this whole point about how, for instance, making the Sydney Opera House in Lego, then you actually can feel like you've seen it if you're a blind person. And it was just really fascinating. And so it continues our arc of Lego.
Yeah, that's a long arc. Hey, Mark, I see you're on the sidebar of my YouTube thing here. Look, Mark Fuccio introduces Drobo. Mac voices.
Oh, okay.
Because I looked at it at some point, and now you're here.
It's Chuck Joyner's show. Great show.
Yeah, it's Mark went on to explain how to set up a Drobo and get apps and so forth. Who Alan, I didn't get you yet, right?
Yes.
Alan,
right.
what do you have?
I have a photography contest. We frequently pick these, but this was a new one on me. This is a link to the finalists in the Comedy Wildlife Photography Awards.
This is so funny.
So you see the dramatic wildlife photography and all the stuff we're accustomed to seeing, and these are the shots that you may not see all the time.
This is a riot.
The bloopers,
yes.
eh?
They're bloopers. They're animal bloopers that are a lot of fun.
There's a great one here of the bird pooping on the buffalo's head.
Yes.
It's great.
I like the Komodo dragon coming out of the toilet, too.
This is just fabulous.
Yeah.
Yes, these are really good.
So I see there's a bear walking upright. I don't know if you heard this, but we used to have one of those in New Jersey. It was well known. And we just went through a bear culling in New Jersey where they let people hunt bear with bow and arrow.
Right.
And they killed 450 bears, including the walking upright bear. Someone shot it.
Petals. Yeah.
It's so cool. He apparently always walked upright. There was something wrong with his paws, or they couldn't figure it out. And they shot him.
Well, he would have been an easy target.
That's pretty cool. Well, I have also a photography site. This is a photographer named Mike Kelly, who does a lot of cool stuff, mostly involving airplanes. And the ones I think are cool are called air portraits. What he does, he goes to different airports and he puts his camera on a tripod and he takes pictures of planes taking off. And then he makes these montages that are just amazing. He'll go to Amsterdam Schiphol or JFK or Dubai, Frankfurt, LA. And he'll have like 200 airplanes taking off of all different types. And they just look amazing. I just thought they were so cool. It's crazy. And then I found also he has a series called Final Approach where he's at the end of the runway under the airplanes. And he has a series of underbody pictures as well, which I always get fascinated when I'm in the parking lot of the airplane and one of them flies over really low. And you could see all the wheels. And oh, my gosh, it's so amazing. And this guy's taken tons of pictures of them. The coolest things, though, are the montages. Those are really, really neat. Yes. Because he's got to get them put in a way that they look natural. It looks like there's just 100 airplanes landing or taking off. Yes. Really cool stuff. I don't remember where I found that, but it's pretty cool. We do have a listener pick from Hannah, who writes, Dear TWiVome, I doubt I'll be number 50, so this was a response to the contest. But I wanted to write in anyway and share some links. I saw an interesting article today about a black widow venom gene that was found in a phage that infects Wolbachia. So that's the source of the article for today. She sends a BBC article in the original link to the paper. I've also got a science pick for you. Raphael from Frozen Flow Glass makes stunningly beautiful borosilicate glass sculptures of arthropods and other animals. Some are fanciful like his Halloween designs, but what I really love and what makes this a science pick are his realistic depictions of individual arthropod species like the black and yellow mud dauber bringing mud back to the nest.
Wow.
Photos of his work can also be found on Instagram.
These are amazing.
Yeah, they're gorgeous. All the best from Berlin, where it is currently a chilly 7°C and cloudy. This is only about a week ago. Yeah, it's cold. So, black and yellow, remember that, everyone. That's how you pronounce my name. Black and yellow, Racaniello. I'm named after a mud dauber. Hannah. Thank you, Hannah. That'll do it for TWiV 412. You can find it at iTunes at microbe.tv/TWiV. And if you have questions and comments, send them to TWiV at microbe.tv. And please consider supporting our science shows, TWiV and all the others. Go to microbe.tv/contribute. And there, I have a whole number of ways, a whole bunch of ways. Let me get it up here on the screen, /contribute, but you can help support us so that we can do different kinds of shows on the road and so forth, go to different places. We have a Patreon account where you could subscribe, and if you give certain amounts of money, you get a mug or a T-shirt. We have some new cool Microbe TV show T-shirts. You can donate over at PayPal, a fixed amount. And our Microbe TV store is at cafepress.com/TWiV. And if you shop a lot at Amazon, I have an affiliates link. If you just use that link to do your shopping, we get a percentage of the item. It goes right to Microbe TV. No additional cost to you. But you do have to go through that link, which is on microbe.tv/contribute. So check all that out, please. I want to thank everybody today. Mark Fuccio from Drobo, thank you for coming and telling us about Drobo and other things as well.
Thank you. It's great to be here. This is a great conversation.
Yeah, we have fun, don't we?
Yes, yes.
I wonder what it's like for people listening in on it real time. It's no different from the final recording, basically, right?
From my sense, well, I don't know. I mean, some of the other ones, seems to be this running joke. Oh, you're going to edit that out, and we'll fix it in post, which never happens. So right. I think probably it's just one long take.
Yeah, yep. You just online, you don't get the beginning and the end, the pre and post. Yeah, the pre and post is fun. Well, our guests, Katreya and Molly, left some time ago. They said they had dogs to take care of. I don't know if they were in their car or what, but we thank them for sitting in. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at alandove.com. You can also find him on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the sponsors of this show, CuriosityStream and Drobo. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.